My name is Dr. Elaine Gee, and I'm the Director of Bioinformatics at ARUP. When I tell others I'm a bioinformatician, I'm often asked, what is bioinformatics? My first response, it's complicated. But more specifically, bioinformatics is an interdisciplinary field that pulls together biology, mathematics, statistics, and computer science to create computational pipelines for dissecting biological data. Now, if that doesn't sound complicated, let me tell you more about its applications. There are a variety of uses for bioinformatics. However, my work specifically focuses on its key role in clinical genomic testing. Unlike other testing methodologies, a high degree of computational support is required to interpret short-read, next-generation sequencing data to generate clinically actionable reports. Said another way, bioinformatic tools are the supporting framework that does the behind-the-scenes work to generate consistent and reliable genetic test results. An NGS bioinformatics pipeline is designed to convert raw sequencing data into medical content by detecting genomic variants, calculating QC metrics, and providing annotations that are interpreted by clinical staff. It takes a team of bioinformaticians to pull all this together by using a variety of off-the-shelf tools or even by generating novel algorithms to bring clinical utility to sequence analysis. This is a highly technical field that requires training in a scientific discipline. Bioinformaticians can have a range of educational backgrounds. For example, on my team, we have folks who have bachelor's and master's degrees in bioinformatics and computer science, as well as PhDs with postdoc and work experience in technical fields such as biophysics, medical informatics, and biology. Even within the field of bioinformatics, there are different focuses between the academic and clinical world. Clinical bioinformatics requires a high level of reproducibility, sensitivity, and specificity for consistent processing from sample to sample. This is difficult to conceptualize, so let's use an analogy to help understand the analytical process further. Let's use the analogy of reconstructing the Odyssey by Homer to better understand the bioinformatics process needed for next-generation sequencing analysis. This book was originally written in approximately 700 BC and has been translated into English many times. Imagine a patient sample as one translation of this epic poem. The first step of the NGS process is equivalent to shredding the book into short multi-character sections with breakpoints randomly occurring within sentences and even within words. Each piece of paper has a variable length and is then copied multiple times, with errors sprinkled into text at random low frequencies. The sequencing step is represented by converting the paper fragments where each letter is translated into a color code for the first 150 characters in each paper section. Typically, a single sequencing run processes many patient samples simultaneously, which would be equivalent to coding many different translations at the same time. In our Odyssey analogy, the first step of the bioinformatics process can be thought of as sorting many different translations of the poem back out from a pile of color sequence paper strips that were indexed by translation version. During sequencing, all translations are color-coded and decoded into a single document. The demultiplexing step sorts out from a single document the original 150 character segments that belong to each translation. Imagine the reference Odyssey is the very first English translation of this poem, and we are realigning a different English translation back to this first translation. There will be different interpretations for syntax, which could lead to skipping words, adding new words, or even alternate spellings. Each 150 character segment of the sample translation is assigned back to a specific page, paragraph, and sentence in this original reference translation. This raw alignment is then polished using proofreading steps that reduce the copies of each fragment and corrects errors caused by translation mistakes. Proofreading is done by creating an error model based on known words in the Odyssey at specific locations, and these words are used to calibrate the rest of the decoded characters. The meat of the bioinformatics pipeline is to call variants, or said in another way, to identify deviations in the sample translation against the reference translation. An example of how this would be done is to collect overlapping neighboring paper strips and leveraging the overlapping characters to rebuild a larger sentence or even paragraph that could then as a whole be realigned back to the reference translation in order to identify variation in characters and words modified, inserted, or deleted. Once a list of deviations are generated, the raw locations, or in other words, page, paragraph, and positions in the sentence, are annotated with information to give it context. These final steps would allow a medical director, acting as the editor, to review the short list of discrepancies between the reviewed translation against the reference translation to understand what changes occurred before it's published.